Hey explorers! Today we're going to learn about something that all scientists use to explore the world and answer questions. It's called the scientific method, and once you know all the steps, you can use it for any experiment you do, whether they're in your classroom or your home. In order for us to learn the scientific method, we made this science fair-worthy display. We're going to learn about each step of the scientific method while doing a really cool experiment, a density column. All science starts with the first step of the scientific method, asking a question. Maybe you're curious about something and you want to know more about it, so you ask a question. For example, what happens when we pour different liquids into a container? Will they all mix together? Will they stay separated into layers? Or will one float while another one sinks? That, explorers, is the question we're going to answer today. Now that we have our question that we want solved, we have to move to step number 2, gathering information or doing some background research. Before we move on to the experiment, we need to know a little bit more about the question we're asking. Here's what I learned during my research. Different liquids have different densities, which is the mass of a unit volume of a substance. What this means is how many particles of that substance are packed into a space. Liquids with higher density are heavier and sink, whereas liquids with lower density float. This information helps us move on to the next step, making a hypothesis. Now it's time for a hypothesis. This is an educated guess on what we think is going to happen when we put the different liquids into our container based on our research and any knowledge that we have going into the experiment. Here's my hypothesis. Out of all of the liquids that we're going to be putting in our container, I think that honey is going to be the bottom layer of our density column. This is because I think that it's the densest and heaviest of the liquids, whereas our oil is going to be lighter and float on top. A good hypothesis begins with the words "I think," and then explains what you predict will happen. Take a second. What's your hypothesis? Walter? Moving on to step 4, conduct an experiment. It's time to test our hypothesis, so we're going to build our density column. You can easily create a density column using materials in your own home. For our density column, we're going to be using honey. We've got rubbing alcohol that we've colored red so we can tell what it is. We've got dish soap. We've got water, also colored so we can tell what it is, and canola oil. You will also need a clear jar or container to build your column in, a spoon so you can gently pour your liquids inside, some food coloring to color any of your clear liquids, and then anytime I'm experimenting or using the scientific method, I love to have a trusty notebook nearby to take notes. If you're doing a school presentation like ours, you can use cutouts, printouts, stickers, markers, and you can add some color to it. In front of me, I have bowls with each of my liquids, a half cup of each one. One at a time, we're going to pour them gently into our container and see how they stack up. I'm going to start with my honey. So we just gently pour it into our container. I'm trying to make sure I don't get any on the sides of the container. And just making sure we let all of our honey drip in. For all of our other liquids, we're going to use a spoon so we can pour the liquid and have it gently land on the honey. This will disturb the layers a little bit less so we don't have to wait for them to separate. Grabbing my dish soap, putting my spoon inside the jar. And you can very clearly see that the liquids are staying separate. So far, we can see that my hypothesis is correct. The honey is thicker and more dense than the dish soap, so it is the bottom layer. Here is my water, which I've colored green. And my rubbing alcohol, which is colored red. And last but not least, my canola oil. Now again, my hypothesis is that the oil will be our top layer. Now that the oil's been introduced, I can see that the layers are trying to figure out which one's going to stack on which one. Now that we've poured all of our liquids into our container, that is our experiment. So now it is time to observe and record our results. Analyzing our data means carefully looking at what happened during our experiment, and then comparing it to our hypothesis. For this, we turn directly to our density column. We can see that the honey is in fact our thickest and most dense liquid. Above the honey is our dish soap layer, the blue, and above that, our green water. Then we have our canola oil, this yellow color, and very topmost is our rubbing alcohol, which means our hypothesis was slightly incorrect, because rubbing alcohol is lighter and less dense than any of the other liquids. This is something I love about the scientific method. Your hypothesis is rarely what's actually going to happen during your experiment. So, once you've analyzed your results, you can go back to your hypothesis, maybe tweak your experiment, and have a look at those results. And you can do that cycle again and again until you figure out the answer to your question. Every time you're using the scientific method, make sure you have a notebook handy. This way you can draw pictures of your experiment or take very detailed notes about your findings. The conclusion to be drawn from our density column is that liquids stack based on their density level. Honey very clearly was the thickest and heaviest of the liquids, whereas the rubbing alcohol, which is this red layer, that went straight to the top because it was the thinnest and lightest. The last step is to share what you've learned during your experiment. Scientists share their knowledge so that everyone can benefit. You can share by writing a report, drawing a picture, telling your class, or even making a video. Here is what I would share about our density column. We tested out the density of different liquids by stacking them in a container. We learned that each liquid has a different density, and they stack based on that density. My hypothesis was that honey would be the heaviest of the liquids, which was correct. And that, explorers, is the scientific method, a really great way to explore, discover, and learn. Now you can use it for any experiment you do, whether you're stacking liquids, growing plants, or launching rockets. Subscribe to Clayton's Exploration Station on YouTube and social media so you don't miss any of the fun in the lab. Plus, you can head to explorationstation.net to find the downloadable worksheet for the scientific method or our other experiments. You can also visit our Patreon page to discover how you can support the Exploration Station. Now you stay curious out there, explorers.
Clayton's Exploration Station.